Okay, so the next chapter is bacteria and archaea. Okay? And you've already began this a little bit in lab, but you can see we're probably like 3 weeks behind lab. We're actually about 2.5 weeks behind where I'd like to be. All right, so we need to catch up. So we're going to go pretty quick through this. Okay? Since you've already done some of this in lab, don't bother copying down this first, probably, I don't know, we're going to skip over about 7 slides and get right into bacteria, prokaryotes in the beginning. The first part of this chapter, in chapter 24, talks about sort of the emergence of cells, okay, and the first cells on Earth as being prokaryotes and where they sort of came from. So, right, after the Big Bang and it cooled down, there was a lot of methane in the atmosphere. After Earth cooled, there was water vapor, and that turned into oceans, right? And this interaction of sort of molecules then allowed for spontaneous production of macromolecules. You end up with lipids, proteins, carbohydrates. Eventually, you get spontaneous formation of RNA, and you end up with sort of the products of cells. Artist rendition of what the early Earth looked like, right? We're going to talk about prokaryotes. They are the most abundant cells on Earth, right? Obviously, you've seen the numerous numbers of bacteria on slides that you get from, just a cheek swab. Outnumber your cells, right? Okay. Some prokaryotes colonize bodies of other organisms. You have a whole host of bacteria that live in your intestines that help for digestion. They provide B complex vitamins to you. They produce methane gas when you have something that's not quite agreeable with them, etc. Okay? They live in diverse environments: cold, hot, salt, high pH, low pH, etc. All right, so they're very well adapted to different environments, etc. Quite a lot today. All right, so here's an example of where they are. All right, you have bacteria that live on your skin. This is a handprint touching a blood agar plate, and then it's left to grow. And you can see the bacteria that have colonized over that time. Nice. Okay. So the first part of the chapter, like I said, is conditioned on early Earth that made the origin of life possible. All right. So basically, there's 4 things you have to understand. Abiotic synthesis of small molecules. What does abiotic mean? What does abiotic mean? Sarah, without what? All right. So the prefix A refers to without or against. All right. What does the root meaning of the root word bio mean? Living. All right. So it's abiotic, non-living synthesis of small organic molecules. So it has nothing to do with cells or the production of something from a living organism. It comes from non-living interactions between molecules, between substances. So that chemistry that occurs, where you're going through general chemistry, all of that is non-biotic. You put something in a tube and in a beaker, and it works and it reacts together. That is abiotic. Okay, there's no living things that are ongoing and breaking things down. All right. So you produce molecules such as methane, all right, and then those methane reacts with water for something else, right? Carbon dioxide and water react together to form sort of carbohydrates, right? Those things join. This is joining of molecules to form macromolecules, right? And then those get packaged into sort of protocells. Lipids are produced. They're hydrophobic. They begin the beginnings of primitive cell membranes. All right, so you have membrane-bound droplets, spontaneous formation of RNA, right? Basically, now there's an internal chemistry that's involved. You end up with the beginnings of separate of outside and inside environments, right? And then you have the origin of self-replicating molecules such as RNA and DNA. All right, all of this are just abiotic productions, right, of how chemicals have interacted over time and then developed things of cells. All right, so here's your organic compound, and then we're not going to use them. All right, so this can go through them. This is a graph showing the number of amino acids in, say, 1953 versus number of amino acids in 10 milligrams of the atmosphere. That doesn't, for the number of amino acids and then the mass of amino acids and what it looks like. Right, and that's based upon the atmosphere itself. So here's your abiotic synthesis of macromolecules. RNA sort of produced spontaneously, right, and then they could, small organic molecules can synthesize when they're on some things, right? So we have this sort of, at early Earth, there's really this sort of melting pot of chemicals that are interacting, right? And based upon lightning, heat, sunlight, right? Energy's being put in, lots of things are changing, chemical interactions are ongoing, and certain things are spontaneously interacting. And that's what ends up giving rise to the formation of cells. All right, so then we get to protocells, right? Replication and metabolism are key. All right, so replication, be able to reproduce, right, and metabolize the things from the outside, right? Methane that's in the atmosphere, carbohydrates that are around, all those chemicals that are around these protocells. What can they use from the outside to do something else on the inside, right? Protocells may have been these basically fluid-filled vesicles that you've seen before. All right, so it's just a separation. And in reality, a protocell is a membrane separation from inside and outside and having two different environments, internal versus external. All right? You know that in water, lipids and other hydrophobic chemicals can spontaneously form a lipid bilayer, right? Yes? You put oil in water, what happens? It separates, all right? So that hydrophobicity allows for a separation. All right? You put lipid in a certain water, it's going to form, you put certain lipids in water, it's going to form a lipid bilayer instead of a mycel, right, which is something else. All right? And then if you can add other stuff, you can increase the rate of vesicle formation, vesicles, protocells, and then you can have simple reproduction and metabolism and maintain an internal chemical. This is really beginnings of what we call protocells. So here's sort of this is an example of trying to reproduce early Earth's environment and what's going on for the formation of cells. And they can show that vesicles performed and could reproduce based upon the chemistry that was likely present in early Earth. All right? And then you end up with self-replicating RNA, and we know what a ribozyme is, right? Right, an RNA molecule that can function as an enzyme. And now this is the beginning of what? What are these RNA molecules replicating going to serve as? They're going to serve as them. This is the beginnings of your genomes. Okay? They're going to serve as mRNA. This is going to be the beginning of genes, right? And then you have natural selection, right? And then the early genetic material, and they base this upon what's called an RNA world. Okay? I'm going through this quick. This stuff is just an introduction to sort of how we've got to no cells to cells, and how all the chemistry that's ongoing prior to that end up. All right? And then you end up with vesicles with RNA. Right? Now RNA's function as a genome. You end up with something that has a membrane, a genome, and some type of metabolism. That's the beginning of a cell. All right? We're going to skip fossil life. What do you know about prokaryotes? Unicellular. Okay. What else? Small. Smaller than eukaryotes. What else?
No membrane-bound organelles.
No membrane-bound organelles, which also means they do not have no nucleus. How is the genome or the DNA structured? In a nucleoid. Okay. What's the nucleoid? Right, it's a region of the cell that basically has the DNA. What else do you know about prokaryotes? All right, so translation and or transcription and translation are linked. Right, they go on. They are ongoing at the same time. What else about their metabolism? Where can they live? What can they do? Some can live in extreme environments. What else? They reproduce quickly. Okay? Anything there? No? Yeah. Okay. Anything else? That's all. They can have flagella. All right? Or cilia. So they can have external extensions of the cell that allow, what are the purpose of flagella and cilia? Movements. Okay? Do they have cell walls? Yes or no? Some yes, some no. No, they all do. Some have extensive cell walls. Some have less extensive cell walls. All right? They come in a variety of shapes. What are some shapes? Circular, spherical, those are cocci. What else? What's that? Like that. Those are what shaped? Those are rods. Those are bacilli or bacillus. Okay? And then you have spirals, which are spiruli or also spirochetes. All right? They can form colonies. All right? So what we're going to go through is we're going to go through their metabolism, sort of some of their properties, and then move on. All right, so here's some examples of your spherical, rod-shaped, and spiral. That's a spirula. All right, so you have different shapes, and then they can be grouped based upon how their unicellular shapes form colonies. So if they form sort of clusters, sort of grape-like clusters, they end up called streptococci, right? And they could, if they are two spheres together, that's diplococci. All right, you have staphylococci, which means they are circular, but they grow in long chains, singular chains instead of branched, etc. Okay, about that in microbiology. All right. So, key feature of nearly all prokaryotic cells is their cell wall. Very few prokaryotes do not have a cell wall. All right? Very few. It maintains your cell shape, boundaries, protection, all right? And also protects it from a hypotonic environment. All right? Eukaryotic cell walls found in plants are made up of cellulose or chitin. All right, but bacterial cell walls are made up of something else called a peptidoglycan. Right? Peptidoglycan meaning what? What does that sound like? Peptide. So it's probably some proteins or amino acid chains linked to what? Carbohydrates. All right. So it's actually sugars that are modified and linked through peptide linkages. All right. Depending upon the layer of peptidoglycan will change and determine whether or not a bacteria is gram positive or gram negative. You did that in lab, correct? Correct. All right. Archaea. What are archaea? What are those different? Why are they different than prokaryotes or bacteria? They live in extreme environments, right? Therefore, they have modified cell walls and proteins. All right. Archaeal cell walls contain polysaccharides and proteins, but do lack the peptidoglycan. So that's the difference between archaea and bacteria. Okay? And that's your gram stain to classify bacteria. Gram positive have simpler cell walls with a large amount of peptoglycan, but gram negative bacteria have less peptidoglycan, but they have an outer membrane. Okay? So gram positive means whether or not that bacteria can retain the stain within the cell wall during the processing. All right? So it tells you how extensive the peptidoglycan layer is. Okay? Questions on that? All right, so here's your gram-positive bacteria and gram-negative bacteria. All right? So gram-positive bacteria have a thick layer of peptidoglycan, one membrane, inner membrane within the peptidoglycan layer, and then the gram-negative bacteria have a thinner layer of peptidoglycan and, however, have two, an inner and outer membranes. All right, questions on that? All right. Peptidoglycan, because it's used for protection, maintenance of cell shape, etc., is a target for antibiotics. If you can prevent a bacteria from producing the peptidoglycan that is going to be needed for reproduction, or let's say maintenance, or for that bacteria to live, you prevent that bacteria from reproducing, right? It weakens the peptidoglycan layer, it ends up weakening the cells, etc. Right? So that's it is a target for bacteria, and actually penicillin is one that targets the peptidoglycan layer of cells. So those antibiotics that target peptidoglycan, are they more or less effective against gram-positive bacteria? Why? So would they be less effective against gram-negative bacteria? Why? Right, because it has a thinner peptidoglycan layer, really doesn't depend upon it. And then also outside of that is an outer membrane that will help protect it. All right? There's also, in many prokaryotes, there's even an outer polysaccharide layer outside of the peptidoglycan layer, which is called the capsule, and it covers many prokaryotes. The capsule is important for attachment, okay? Attachment to surfaces, attachment to other bacteria, etc. All right, so for interactions. All right, so here's a lovely bacterial cell wall. You think that's likely a gram-positive or gram-negative bacteria? Gram-positive. Why?
It's pretty thick.
It's a pretty thick cell wall. All right? And then outside of that, you see that polysaccharide chains, sort of the fuzzy stuff around it. That is a capsule polysaccharide. Again, attached for attachment. And what you see up there, you see over here, is a tonsil cell, and this bacteria is attaching to and holding on to the epithelial cell membrane. All right? Outside of that, some bacteria develop resistant cells called endospores when they lack an essential nutrient. Okay? Anthrax. What is anthrax? It's a disease, right? Caused by what? Who did a report on anthrax? Cody. Anthrax is a disease caused by what? Say it loud and proud, son. a rod-shaped bacteria known as what? Bacillus anthracis. Okay? How does one contract, Cody? How does one contract anthrax?
Inhalation.
Inhalation of what?
The spores.
The spores. Okay, so Nick develops a very sick sense of humor and decides he wants to overthrow the government, which I don't put it past him, by the way. And he decides, I'm gonna infect all these congressmen with anthrax. Right? This also actually happened, right? Send letters with this white powdery substance. It wasn't Nick. It wasn't Nick. Okay? Or for this white powdery substance to the offices of all these congressmen, they open it, right? Inhale. The spores, right? Those spores are endospores. They come from cultures of Bacillus anthracis, which then have been dried and desiccated. The biology of the bacteria recognize that there's less nutrients, less water around them, and then develop this sort of dormant stage, all right, and become endospores, right? Those spores stay, they contain genetic material, polysaccharides, etc., right? Now those spores are inhaled, what does the lung and the respiratory system serve for that endospore and that bacteria? As a what? An environment. A good environment for reproduction. Spores become activated, right? The Bacillus anthracis bacteria begin to reproduce and cause problems. Okay? So that whole production of endospores is, again, something you go through in microbiology. Right? Other bacteria, so that is really a dormant stage of a bacterial cell. But why are we talking about it in terms of outer covering? It's an outer shell that actually helps protect the inside material of the cell. All right? Other bacteria have what's called fimbriae. All right? They're sort of these finger-like projections that allow for help for attachment. All right? Stick to substrates, attach to other individuals. And then there's pili, or sex pili. All right? Pili are like a tube, not just say a filament. There's actually a tunnel that can be attached between two cells. Right? And that allows for an exchange of genetic material. Right? So there's different functions for all of these. Here's fimbriae. This is E. coli cell. What you see in light purple is the DNA. What you see in sort of purple or the darker bluish color is the cell membrane, the cell wall. All right? And then you have in yellow and pseudo-colored are fimbriae. Right? It allows for attachment. How do prokaryotes move? Flagella, cilia. What causes them to move? Why would they move? So, movement or cellular motility is also known as taxis. All right? Taxis. And so there's usually, why would a bacteria move from one place to another? For nutrients. All right, it's going to move from in order for nutrient to move where there is an abundance of nutrients, to move away from there is less problems, all right, or other less problems. I will say less lack of nutrients, or the presence of something that it's not going to be allow it to live, type thing. All right. So taxis, the movement, the ability to move toward or away from some type of stimulus. In the case of nutrient movement, that's called chemotaxis, right? Whether or not there's a chemical that's going to stimulate movement of that cell toward or away from something, all right? Some prokaryotes exhibit phototaxis. What is phototaxis? Moving towards or away from light. All right. So if a photosynthetic prokaryote, would it move toward or away from light? Right? So that would be positive phototaxis, moving toward light. Okay? And most motile bacteria propel themselves by flagella. You all know what a flagellum is, right? It's an extension of the cell, whip-like motions which will drive and produce movement and move the cell in a certain way, all right? The flagella of bacteria, archaea, and eukaryotes are composed of different proteins and likely evolved independently, all right? So the structure of them is somewhat similar, okay? But their constituents, what they're made up of, is quite different. So this is a flagella of an E. coli cell. All right, there's rods. It extends from the cell, the cytoplasm, the inner cell, across the peptidoglycan layer and across the outer membrane. All right, there's a couple different important proteins. You have the hook, the filament, and the motor. All right, the motor proteins allow for rotation. Okay, the hook protein is an attachment to the filament. Okay. Filament is the extension of the protein of the flagellum itself. The purpose of the motor is to rotate the hook. The hook rotates, and then the filament goes with it. All right. There's a whole. What are flagellum composed of in eukaryotic cells? What protein? What part of? Extensions of centrioles, right? 9 plus 2 centrioles. Remember that? That sound familiar to you? No. Vague? It failed. Okay? So your motor, hook, and filament. So there's modified versions of proteins that perform other tasks. Right? It likely evolved as existing proteins were added to the ancestral secretory system. What does that mean? Don't worry about it. Right? It's just showing the evolution of it, where it came from, right? And this is an example of exaptation, where existing structures take on new functions, all right? So they're saying that in some way there was some function of the cell that the flagella proteins did, but then they changed their function over time. All right? We'll stop there. Any questions? These slides will be posted this afternoon, okay? I'm going to continue to go through this pretty quickly. All right, there's like 120 slides in this chapter. I want to get through this by the end of next week. Mm-hmm. Yep.
It says I got 50% on all the quizzes, but like I know I didn't. I would have remembered breaking out that, but I didn't.
Okay. Okay. I'll check again.